Without further ado, I'd like to introduce our speaker for today. Today we have Abhilasha that's going to be presenting. She has a master's degree in chemistry with an extensive research experience in molecular biology techniques and methodologies for application in genomics and transcriptional profiling. Her area of research primarily focused on clinical and translational research, and she currently works as a senior technical support scientist at Novogene. So we're excited to have her present today. And Abhi, if you'd like to take over, we're eager to see what you have to share.
Thank you, CB, for your introduction. And I would like to thank everybody for joining today's session. Let's get started. Today's topic is eukaryotic mRNA-seq sample preparation and getting qualified total RNA samples for our RNA-seq. So the content of this topic has been divided into 4 parts. The first part is eukaryotic mRNA-seq introduction, and the second part is sample quality control, and third part is sample preparation for eukaryotic mRNA sequencing, and the last part is the frequently asked questions for submitting samples for RNA-seq surveys. So in the first part of our mRNA-seq introduction, we will discuss about the basics of RNA. Sorry, it slipped my. We'll discuss about the basics of RNA, the types of RNA, Novogene's Falcon platform, the applications of mRNA-seq surveys, and mRNA-seq workflow at Novogene. So as we all know, the RNA is a single-stranded nucleic acid polymer of 4 nucleotides, adenine, cytosine, guanine, and uracil, joined through a backbone of alternating phosphate and ribose residues, converting the information from DNA into proteins, or some serve direct role in cells like ribosomal RNA and transfer RNA. We all know the concept of central dogma, the flow of genetic information from DNA to RNA to make functional products as proteins. DNA undergoes the process of replication. Replication is the process by which DNA makes a copy of itself during the cell division. When DNA unwinds, when DNA unwinds, the transcription process takes place. The process by which the DNA is copied to RNA is called transcription. And the reverse of it is called the reverse transcription, where we generate a complementary DNA from RNA strand. This process is called reverse transcription. This process happens, the transcription process happens in the nucleus of the cell. Once the RNA are made in the nucleus, and then they are exported into the cytoplasm, with the translation machinery happens, the machinery that actually makes proteins. The process by which the proteins are synthesized from the information contained in a molecule of messenger RNA is called translations. Proteins play a very important role in our body. They help, the key functions of proteins are structural support, cellular mechanism, immune-related and enzymatic reactions, and many more. So this part of the slide is talking about the different types of RNAs that we have. So the Human Genome Project has led to the discovery that about 80% of our DNA is transcribed in RNA molecules, but only 2% of the human genome is translated into proteins. The rest mostly is produced molecules called non-coding RNAs, which are heterogeneous class of RNAs involved in different steps of gene regulation. They've been classified according to their length as well as according to their function. They are classified according to their length into small non-coding RNAs as well as long non-coding RNAs. And according to their function, they are categorized as regulatory non-coding RNAs as well as housekeeping non-coding RNAs. Their involvement has been widely demonstrated in all cellular processes, as well as their dysregulation in human pathologies. So let's talk about the coding RNA part, which is the messenger RNA, also abbreviated as messenger ribonucleic acid, is a complex high molecular weight that functions in cellular protein synthesis and also replaces DNA as a carrier of genetic code in some viruses. The long non-coding RNA falls under the category of linear RNA, and most of these long non-coding RNAs are heterogeneous class of non-protein coding RNA with a length of longer than 200 nucleotides. Under the small non-coding RNA, we have microRNA, snRNA, which is small nucleolar RNA, snRNA, which is small nuclear RNA, siRNA, which is small interfering RNA, piRNA, which is pi-way interfering RNA. Small non-coding RNA are about less than 200 nucleotides in length. The microRNA in the small non-coding RNA part plays a very important role. They are about 18 to 25 nucleotides long and function in gene regulation in most eukaryotics. A microRNA can inhibit or silence the gene expression by binding to target mRNA and inhibiting translation. So thereby they prevent the function of proteins from being produced. So hence microRNA plays a very significant role in cancer as well as many disease related studies. So unlike the linear RNA, we have circular RNA. The circular RNA are formed by covalently closed continuous loops and act as a gene regulators in mammals. Circular RNAs are formed or produced by non-canonical splicing event called back splicing. They also bind with microRNA and make sponges, as you see in the picture, that prevents the microRNA from binding to their targets. Under the housekeeping RNA, under the housekeeping non-coding RNAs, we have two kinds of RNAs, that is transfer RNA and the ribosomal RNA. They are expressed in all cell types and carry out essential functions in the cells. We at Novogene offer several different services for RNA-seq. The main service is the messenger RNA-seq service. If you are interested in our messenger RNA-seq service, we do offer that as a service, as well as we do offer long non-coding as a service, microRNA as a service, as well as circular RNA as a service. If you want to look at these individual services, we do offer them as an individual services. But also, if you're looking for the whole transcriptome service, all of this is included under the one package, and we offer that as a whole transcriptome sequencing service. Please do reach out to us, like the Novo team or your dedicated technical support, if you need more details of all the services that we provide for RNA-seq. So let's just talk about, again, the RNA-seq part and like the Falcon system that Novogene has. So as we all know, the mRNA, the messenger ribonucleic acid, is a single-stranded molecule of RNA that corresponds to the genetic sequence of a gene and is read by a ribosome in the process of synthesizing a protein. Powered by the Illumina platform, Novogene's eukaryotic mRNA sequencing includes the measurement of all the mRNA transcripts in a sample at a given time. It enables profiling, gene expression profiling, as well as exploring the gene function, as well as gene structure. The picture that you're seeing at the bottom is the Falcon platform at the Novogene. Novogene's Falcon platform is a breakthrough in traditional NGS field. And Falcon is an automated sample storage system integrated seamlessly with an intelligent delivery system. Falcon provides customers with an intelligent and efficient and a robust service for their sequencing needs. Falcon, as an intelligent delivery platform, further can optimize the NGS procedures, which is also designated to avoid any manual errors and also eliminates the collaboration of islands in NGS operation. Falcon enables intelligent operations from sample extraction, sample QC test, library preparations, library quality control, library pooling, and all the way to the bioinformatic analysis. Falcon guarantees Falcon guarantees the cycle of a large project of processing about 2,000 to 3,000 samples in 24 hours, reducing the production time, the TAT, the turnaround time by 60%. Let's just talk about some of the applications, as RNA-seq has been widely used in many fields. So we can apply RNA-seq in medical research as well as agricultural research. In medical research, we can do in pathological mechanism. Our BI analysis includes the oncogene and fusion gene analysis. It can be studied in tumor subtypes classifications, looking at molecular markers, drug targets, where we can look at the gene expression variations in the drug versus treated samples, human evolution studies, clinical diagnostic studies, personal health care studies, where we can look at disease-related gene analysis, including oncogene and fusion gene analysis. It's applicable in the agriculture research field, such as it can include the development and adaptability studies, crop evolution studies, and ergonomic trait studies. So with our BI analysis package, we can look at the gene expression and functional analysis between ancestors and offspring. We do offer several different BI analysis packages, the standard and the quantification. Please do reach out to us if you have any custom analysis that you're interested in. We evaluate project based by case, and we get to meet your project needs. Oh, this is a slide showing about the workflow from start to end at Novogene for the mRNA-seq surveys. To ensure the successful RNA experiment, we expect the RNA to be of good quality and quantity so that the library construction and sequencing are successful. So upon receiving the total RNA from you, we QC the sample, both quantity as well as quality, and we evaluate sample as a pass, hold, or fail based on our QC criteria. We send clients a detailed QC report with all the information about your sample QC parameters, and client gets to review all the details and provide with us with a confirmation to proceed with library prep and sequencing. Once we receive a confirmation from you, we proceed to the next step called library construction. The picture that you see below is the standard library construct, which consists of a P5 and a P7, the oligos that bind to the flow cell, a read-one sequencing primer. It has a read-to sequencing primer and a DNA insert as well as index for demultiplexing purpose. Once the library construction is done, we QC all the libraries, and based off of the qPCR values, we sequence on this NovaSeq or platform. The sequencing strategy, our standard sequencing strategy is PE150, it's just paired in 150. Once the sequencing is done, depending upon the client's need, whether the client wants Novogene to handle the BI analysis, or if clients would just want the raw data files, we can either do a without BI analysis project and a with BI analysis project. So for without BI analysis project, we release raw data in the form of FASTQ files. For the clients who choose Novogene to handle the BI analysis, we have a bioinformatic analysis. We offer two main analysis pipelines. One is the quantification analysis package, and the other one is the standard analysis package. the quantification analysis is looking at the differential gene expression, and the standard analysis includes differential gene expression and also looking at the structural analysis as well as enrichment analysis. So depending upon your need, please, you can either pick a quantification package or a standard analysis package. If your needs to be customized, if you have a customized analysis that you're interested to do, please reach out to us. We will evaluate that and we will get this to be done. So the second part of the topic is the sample quality control. In this session, we will talk about the instruments that we use for testing the QC, the sample QC, as well as sample requirements if you're submitting tissue samples or the non-tissue samples. So as you all know, Novogene is a global company. We have labs in US as well as China lab. The Davis lab is the US lab, and in Davis lab, we use qubit preliminary to test the QC, a total RNA samples for RNA quantity, and we use Agilent bioanalyzer to check the RNA integrity as well as the quantity. In China lab, we use nanodrop for preliminary QC to test the RNA quantity, as well as we use the Agilent 2100, also another platform called AATI, which is an Agilent 5400 to check the RNA integrity and quantity. We will talk more about the quantity and the quality parameters in the next slide. So AATI stands for Advanced Analytical Techniques, which is acquired by Agilent. It's an Agilent 5400. It's a fragment analyzer, and it's used as an automated parallel capillary electrophoresis principle. It works on the automated parallel electrophoresis principle, and it's used for nucleic acid QC analysis, and it is a fully automated for a wide range of molecules. We can use a quantitative kits to analyze RNA, genomic DNA, small RNA, next-gen sequencing libraries, as well as large fragments. Currently, we are using this platform in our China lab for all non-directional libraries, and the sample needs to be greater than or equal to 50 nanogram per microliters to use this robust platform for QC check. Here is a table talking about the different sample sources that we accept, as well as on the top you see different kinds of library preps that we offer and the sample requirements. So for the cell pellet samples, we recommend submitting greater than or equal to 5 million cells for the non-directional library, as well as directional library, mRNA library, as well as ribosomal depletion library. For the whole blood samples, we recommend submitting greater than or equal 5 ml across all the 3 different library types, that is the non-directional mRNA library or directional eukaryotic mRNA library, as well as for ribosomal depletion approach. For the flesh plant tissue, we recommend submitting greater than 500 milligrams across all the micrograms across all the 3 library preps that we offer, as well as the fresh animal tissue is greater than 300 mg. For the thalle samples, which are the vegetative masses from any kind of drivers groups like algae and fungi, we expect you to submit either greater than or equal to 7 million cells or greater than or equal to 4 ml. For stool samples for metatranscriptomic library prep, we expect you to submit greater than or equal to 500 mg. So if your samples doesn't fall in these criteria or your samples are very less, please do check with us, and our labs has experience working with lower amounts too. This is another slide talking about if you're trying to submit total RNA, which is the non-tissue sample for RNA-seq service, then here are the amounts for different library types that we offer. So for total RNA for non-directional library prep, we recommend submitting greater than or equal to 0.4 micrograms and a volume of greater than equal to 20 microliters, or RIN values of greater than equal 6.8 for animals and greater than or equal to 6.3 for plant and fungi, and a concentration of greater than equal to 20 nanogram per microliter. And we expect the purity to be O260, 280, as well as O260, 230 ratios to be greater than equal to 2 with no degradation or contamination. For total RNA for directional mRNA libraries, we expect you to submit a greater than or equal to 0.8 or 800 nanograms or 0.8 micrograms. The rest, the volume, the RIN, the concentration, and the purity, the standards remains the same. If you're submitting total RNA from the blood samples for globin clear or globin 0, so we expect you to submit greater than or equal to 0.8 micrograms, and as well as greater than equal to 20 microliters. The RIN value needs to be greater than or equal to 6.8 and a concentration of greater than equal to 20 nanogram per microliter. We Novogene also accept double-stranded, amplified double-stranded cDNA as the starting material. We do not accept single-stranded DNA. We only accept double-stranded cDNA as the starting material. For when you submit, so we recommend you submitting greater than or equal to 100 nanograms of double-stranded cDNA at a volume of greater than or equal to 20 microliters and at a concentration of greater than or equal to 10 nanogram per microliter. If you're submitting double-stranded DNA, we recommend or we encourage clients to run their samples on Agilent Bioanalyzer or a tape station to look at the fragment size distribution. So we expect the fragment distributions between 400 to 5,000 base pairs with the main peak at around 2,000 base pairs. And we expect the OD 260 to 30 to be greater than equal to 2 and no degradation and contamination. We Novogene also accept for samples like, for low input and ultra low input service. For total RNA for ultra low input mRNA-seq service, anything between 100 to 400 nanograms will fall under our low input mRNA-seq service, and we expect client to submit greater than equal to 100 nanogram, and the volume, the RIN, and the concentration remains the same to the standard RNA-seq pipeline. For anything which has below 10 nanogram per microliter, the total amount falls under our ultra low input mRNA-seq service. If your quantities are below and the quality is below the recommended, please do check with us. We do case-by-case evaluation and give you feedback accordingly. The third part is sample preparation. In this, we will talk about what are the factors that Novogene is considering and evaluating your samples to be whether passed, failed, or hold, and what precautions we need to take while handling either tissue or total RNA samples, and how are we supposed to handle like tissue samples when you're doing homogenization, or what precautions we need to use, we need to follow. And we will talk about the abundance of RNA in different organs, as well as the proper tissue preservation methods. So when we talk about the low quality RNA, it's at Novogene. We are not talking about the quality of RNA. We at Novogene has been deeply involved in NGS for a decade and has accumulated experience in processing samples in the RNA-seq project. We conducted in-depth research on low-quality RNA samples and found that Novogene low-quality RNA is mainly caused by the insufficient amount of RNA degradation. As you all know, based off of the initial QC criteria, we evaluate your total RNA samples to be either pass, failed, or hold. Any sample that has been given a pass means technically the sample has passed, has good quality as well as quantity of RNA. Any sample that has been evaluated as hold or failed. The three main factors that are causing to evaluate your sample as hold or fail is because of the insufficient amount to start with, or because of the degradation or presence of other impurities. Based on our experience and the X number of projects that we have worked, it looks like the samples are evaluated either pass or fail. The most of the time, the 60% of it is because of the insufficient amount of RNA to start with, and 30% of is because of the low quality, which is the degraded RNA, and the 10% of is it because of the impurities that are present in your total RNA samples. Also, I would like to make a note here is any sample that is evaluated as a whole, in our experience, would have even though it's evaluated as a whole, because that's our QC criteria and our QC metrics are pretty stringent. But any sample that has been evaluated as whole can still yield a good library preparation success rate for at and about 98%. And in general, the data quality of this whole sample is enough for analysis in most projects. But for the samples that are evaluated as fail, the data quality would be affected more by the RNA degradation. So we recommend clients, if it is possible, to submit a replacement sample in the samples that are evaluated or fail, that are evaluated as failed. So these are the few things when we are preparing how to prepare high quality RNA samples. We do have some recommendations and suggestions. The first and the important thing is prevent degradation that is caused by exogenous RNases. As we all know, RNases are a large group of hydrolytic enzymes that degrade ribonucleic acid molecules. So we need to make sure be free from the exogenous RNA contamination. And the degradation that is caused by this exogenous RNases is one of the most common risks during the sample preparation. So we have listed a few things up here. So let's just talk one by one. So the experimental reagents, so we want to point out, make sure the reagents that you are using to process your RNA samples, make sure they are not expired, or they've not been opened for a while, or they've not been sitting on the shelf for a long period of times. So make sure you use the the good reagents for your RNA extraction procedures, as well as the environment, the lab space, or the hood that you're working. Make sure you decontaminate thoroughly prior you start your RNA extraction procedure or RNA experiment. Short operating procedures, we suggest not to operate for prolonged period of times, and this will cause some RNA to degrade. So we recommend suggesting to operate in short. Suggesting to limit your operating time, means to decrease the risk of RNA degradation. And also we expect the laboratory personnel to follow this SOPs. Optimize the SOPs based on your study needs or the sample types, and work accordingly. All the laboratory equipment, such as the pipettes or tips, any utensils that you use for your experiment or for the RNA extraction, make sure they are very sterile, and make sure sometimes if they are ready to available RNase-free equipment. Some more points about to prevent the degradation that caused by endogenous RNases, especially when you're working with the tissue samples and those of that are very rich in RNA endogenous enzyme-rich organs such as spleen and thymus. They're very easy to degrade. So we recommend putting all the tissue samples on dry ice before crushing them. And also crush the tissue samples under liquid nitrogens. We recommend doing that. And also use more lysate during the homogenization process. And also when packing the samples, please try to prevent the contamination and degradation. And also please do check Novogene's tissue sample requirements prior you sampling out your tissue samples if you're planning to submit the tissue samples for RNA extraction. So please confirm with us the tissues amount and the amount, how you're supposed to pack them, or how you're supposed to freeze them or snap froze them. So please do check our Novogene extraction guidelines guidelines for proper sampling about the sample amounts, tissue amounts. And also, if you after extraction, the RNA should be packed separately as placed in frozen storage. Please avoid freeze thaws to avoid any kind of degradation. So here is a table, and so as we all know, different organs have different RNA abundances. So try to choose high abundance organs such as liver, spleen, and heart for RNA extraction. If you need to use parts that are not rich in nucleic acids, so you could increase the tissue amounts appropriately. Here is a table showing a different organs with a different abundance of the RNA. So we have the sample source as the fresh animal tissue. So in high abundance, the organs for a liver, spleen, pancreas, and heart, it's about 2 to 4 microgram per mg, whereas the medium abundance organs are brain, embryo, kidney, lung, thymus, ovaries. So the RNA abundance in these organs is 0.05 to 2 microgram per mg. And the low abundance organs are bladder, bone, fat, and they have like less than 0.05 microgram per mg of on the animal tissues. Accordingly, in the fresh plant tissue, the high abundance organ is the blade, and and the RNA abundance is about 0.2 to 0.3 microgram per mg. And the medium abundance organ is the rhizome, and the RNA abundance is 0.1 to 0.2 microgram per mg. And in the low abundance category, it's fruits, flowers, and seeds. So in these, they have less than 0.1 microgram per mg. But again, if you have any questions related to the sample submissions and the sample, like the amounts, please do check our website, which has more details about the tissue submission as well as RNA submission details. And and also choosing the proper tissue preservation is also very important. The preservation method used is also an important factor affecting the RNA quality. So here is the list that we are showing you, like the different tissue samples and the processing type and the preservation that we highly recommend, strongly recommend and do not recommend. For animal tissues, if you prefer like grinded tissue, we highly recommend you to snap frozen them, and we do not recommend dissolving in RNA later. If you're planning to ship ungrounded tissue, then you can either we strongly recommend snap frozen, and also you can dissolve the tissue in the RNA later. For plant tissues, fungus, and bacteria, you can highly recommended is snap frozen. We do not recommend dissolving in RNA later. For blood sample, for the whole blood processing, we recommend submitting the packaging tubes and also the snap frozen blood, but we do not recommend dissolving in RNA later for the blood sample. The cell pellets, we always recommend you to, and it is strongly recommended to submit them as snap frozen and do not dissolve in RNA later. So, and also frozen materials should be stored at -80, and please avoid repeated freeze thaw cycles and keep at a minimum as they enhance nucleic acid degradation. Some more points that we are talking about, like labeling and shipping samples. For shipping samples, make sure you make well arrangements. We recommend, using parafilm to seal each and every tube, because parafilm helps to prevent leakage during transit. If it is like less number of samples, like say less than 24 or 10 or 12 or whatever, you can place the tubes in 50 ml conical tubes, and then, or you can use any box depending upon what you have, and use some cottons or absorbent papers to prevent the tubes from moving around inside the containers. But if you have more than 24 samples, or if you're planning to submit more number of samples, it's better to use some cryo boxes with an organizers and seal those boxes or tubes in Ziploc bags for safety. And also, we always recommend shipping samples on dry ice And we recommend using 5 lb of dry ice per day and at least a 10 lb of dry ice for overnight shipping. So the last part of the topic is these are some of the frequently asked questions for submitting samples. So here is the first question that we have is, can Novogene accept extracted RNA in RNA later? The answer is no, because that you have already extracted your RNA. So we recommend you to elute your RNA in either the elution buffer that came with the commercial kit, if you're using any commercial kit, or the nucleic RNase-free water or double distilled water. If you put your RNA that is extracted in an RNA later, we need to re-extract the RNA, and we cannot guarantee, because this could cause the huge sample consumption and leading to the waste of precious samples from your end. So we strongly recommend not to dissolve your total RNA in any reagents like RNA later or triazole. Please elute your samples in the elution buffer or water or double distilled water. Next question is, can Novogene accept RNA samples dissolved in ethanol? Yes, we do accept RNA in ethanol, but RNA needs to be reprecipitated and eluted back. So there will be an extra fees to do this. So however, we again do not recommend client to do so, since RNA purification can cause great loss of the sample amount. Please check with the TS or the Novogene team what is the best way to submit your total RNA samples, in which form and which elution buffer, or what are the questions that you have. Okay, the third question is, can Novogene accept the RNA samples preserved in RNA stable or Genpregra RNA? Yes. The answer is yes. We do accept the RNA in the lyophilized or like the pellet or the powder form. You need to follow the kit protocol, the RNA stable, which is from Sigma, or the Genpregra. Please follow the manufacturer protocol for pelleting the RNA. Once we receive the RNA pellet, we resuspend in a buffer or nucleus-free water and perform the QC. So we do accept RNA in the pellet form or the powder form, and we do not charge any extra fees for resuspending your pellet back in the RNase-free water. The next question is, does Novogene charge if the RNA sample concentrations are too high or needs dilution? No, we do not charge any extra for diluting your high concentrated total RNA samples. But if you believe that your samples are too concentrated, based on the sample input requirements that we have, you can dilute your samples. Or if you do not want to freeze thaw your samples, you can just submit them as is to Novogene, and Novogene will dilute accordingly and perform the QC and use it for downstream processes. And there is no additional fee to do this. Thank you everybody for joining today's session. Thank you, Charles and Marcy, for being the panelist for this session. And I would like to thank Novogene supervisors and Novogene team for giving me this opportunity.
Awesome. Thank you, Abhi, for that presentation. As mentioned before, we are going to go into a live Q&A session where Abhi will be answering questions that you have from this presentation. Like I mentioned before, please do post your questions in the Q&A module here in Zoom, not the chat, as our panelists will be monitoring the Q&A module, and Abhi as well will be monitoring the Q&A module and not the chat. So if you could put your questions there, we'll give it a few moments to allow for everyone to be able to put their questions there. Any questions that we are not able to answer, we will reply and provide an answer for in our follow-up email that has the webinar slides as well as a recording to the webinar, as mentioned earlier. Just a quick reminder, please follow us on LinkedIn, Twitter, Facebook, Instagram, or sign up for our newsletter for cool promotions we're doing, upcoming webinars, and just to stay up to date in what we have in store for you guys and what new developments are there. So at this time, I'm going to give it a few moments real quick just to let some questions come in. Once that question comes in, we can answer those live. So Abhi, there was a question, give me 1 second, around if, from Priya, where if a client wanted to run an RNA from a cell population, how many cells of that type should they send? Could you speak to that a little bit more?
Sure, definitely. As you have seen, one of our slides says we recommend submitting greater than or equal to 5 million cells. So for successful RNA extraction. So, but again, if you cannot get that number of cells, please do reach out to us. We will get in touch with our labs to see if we can accommodate the sample extraction or not. And it's a case by case. So, but our standard recommendation for submitting cells is greater than or equal to 5 million cells.
Okay, good. That does clarify some of that. I think that is a common question. Just redirecting some questions to the Q&A module. Again, if you do have any questions, please do post it in the Q&A module so that we can answer that for you. There was another question actually from Felipe. Abhi, maybe you can speak to this as far as how many replicates do you recommend for differential gene expression experiment?
We recommend at least a 3 to 4 biological or like replicates or per condition for differential gene expression. If you have more, which is good, but because it's per sample price for mRNA-seq, so definitely the minimum is 3 to 4 biological replicates.
Okay, so that's good. That does clarify that. I know that we've had some of previous projects that had to be restructured based on that. So thank you for that clarification. Let's see. If you have questions, please do post that in the Q&A module and not the chat so that we can see that and answer it. Let's see, we have time for about 1 to 2 more. Abhi, there is this question from Marina where she's asking what are some of the sample requirements and recommended extraction procedures for circular and long non-coding RNA. Could you speak to that?
Yeah, sure. So both of the services, they fall under, if you're looking at the individual service, we offer long non-coding as a service individual. The circular RNA falls under our whole transcriptome service. So for long non-coding RNA, we recommend submitting about greater than or equal to 800 nanograms of total RNA as starting material. For whole transcriptome, I think it's maybe 2 micrograms of RNA. Because the whole transcriptome service includes messenger RNA, looking at messenger RNA, long non-coding RNA, microRNA, which is small RNA, as well as circular RNA. So we do not have a dedicated service just to do the circular RNA, but the circular RNA is part of our whole transcriptome sequencing service. So for the extraction, again, the sample requirements are as listed on our Novogene extraction guidelines. That for the tissue, greater than 500 mg equal to, but anything lower, again, we need to do it by case. Okay.
Yeah, I think that one of the common themes that we want to communicate to everyone is that we understand that your project is unique, and although we do have general guidelines, if you have any questions, we'd be more than happy to look at it on a case-by-case basis and make recommendations on how we can make sure your project is successful. So please do reach out to us if you have any questions regarding your specific project, and we can give you some very specific feedback and go from there. So we have time for one more, and just to remind everyone, any question that we have not been able to answer, we will answer in our follow-up emails, which will have the link and the link to this webinar that's being recorded and the slides as well. So we will make sure we answer all these, but we only have time for one more. And Abhi, there is a question from Mickey that's asking around the question of Trizol. Was that recommended? And is there an extra fee for samples extracted using Trizol? Could you speak to that?
Yeah, sure. So what I was trying to say is, yeah, you can, the tissues can be, you can use Triazole for extracting your tissue samples. It's just what we meant to say was, like, after you extract the total RNA, you know, we had a question from clients, can we put it back in Triazole? No, that was the no answer was no. But yeah, you can use Triazole for extracting the tissue samples as a Triazole reagent for extracting the tissue samples. But after you extract the total RNA, we recommend you to elute in elution buffers or nucleus-free water rather than putting again back in our Triazole reagent. So that was the thingy. There's no extra fees if you want to extract samples using Triazole. There's no extra fees for doing that.
Okay, I think that's a good question, and one of the questions that we do get quite often. So I'm glad that we were able to clarify that. Again, that's all the time we have for our Q&A today. I want to thank everyone for joining us. We'll leave the Q&A module open for a little while longer, just a minute or two, so that you guys can post your questions there. Again, we will be able to answer anything that we haven't been able to answer in our follow-up email after this webinar is over. So if you do have any questions, please do post it in the Q&A module. But at this time, we are done with our webinar. Thank you again for joining us. Yeah, we look forward to interacting with you soon. Bye-bye.
Thank you.